This video is about how to estimate size and scale in cell images, or how to solve bacterial ruler problems. The bacterial ruler has a series of scales of biological things, starting with something you can see, a hair, and in powers of 10 going smaller from a hair down to individual molecules. Using bacterial ruler problems to estimate size and scale in images helps them get a sense of what the real sizes are of the structures that they look at, think about, and work with in biology. If you haven't practiced drawing the bacterial ruler, and if you can't reproduce the bacterial ruler on paper or in your imagination, then find my bacterial ruler video to learn and practice. You should practice until you can close your eyes and go through the entire bacterial ruler easily and fast in your mind. It's when they do bacterial ruler problems that it really starts to stick well and it actually becomes useful. So what's our goal? You should be able to look at any image of biological molecules, cells, or organs and make size estimates for anything in the image with no size scale, with nothing telling you what the sizes are in the image. And your estimates can regularly be within 1/2 of the actual size to 2 times the actual size, and nearly always accurate within a factor of 4. When you've done enough practice, your mind will automatically hold proper scaling at all times when you think about biological structures. Many problems and exam questions in biology depend upon size and scale. You'll make fewer errors just by knowing the bacterial ruler and these problems. This shows a pituitary gland, the big thing in the middle that looks vaguely like a heart, in a frog. In the upper left, labeled ME, is the base of the brain. IS is the stalk of the tissue connecting the pituitary to the brain, and NL, PI, and PD in the center, top, middle, and bottom, those are the different parts of the pituitary gland. And over on the right is MB, another part of the brain, the midbrain. But how big are these structures? In this case, the figure legend just states times 150, meaning it's magnified 150 times. But that's not very helpful when I've changed the size of this image. So our goal is, you should be able to look at this and understand there's a series of 4 steps you need in order to solve bacterial ruler problems. Step 1 is there anything from the bacterial ruler in the image? Step 2, whatever you recognize from the bacterial ruler or that you just remember, you can use to estimate size. When you have a size for that one thing in the image, you can use multiples of it or fractions of it to estimate sizes of anything else. Step 4 is, if possible, check your work. Does your answer make sense? If we wanted to start estimating sizes on this image, what would we do? Well, you know that this is part of the brain and a gland. Brain and glands are made of cells. So there might be something in here that's a cell. In fact, with a little practice looking at these, you'd quickly recognize that these dark dots are probably nuclei. And the paler stuff around it, that's the cell cytoplasm. So at the blue arrows, I've circled a couple of things that are nuclei. And now you have something you can connect to the bacterial ruler. Each nucleus has to fit inside a cell. Therefore, a nucleus is smaller than a cell. In the bacterial ruler, a typical animal cell is about 10 microns. So maybe the nuclei are about half the diameter of a cell. That would make each nucleus about 5 microns across. That's the distance between the 2 blue lines. What's the thickness of the stalk at IS in the top left, where the arrow is moving? Now we need multiples of our nucleus, and I'll move them up and over to line them up on the stalk. 1, 2, 3, 4, 5, and you can see that the thickness is about 4 nucleus units, about 4 times 5 microns, or about 20 microns. Let's try another problem. What's the size of the whole pituitary gland? That's the NL-PI-PD. We can duplicate lines, and if you counted these lines, it's about 22 or 23 of the size units of the nucleus. If you measured it from top to bottom, it would be a little more, around 30. So 5 times 22 is about 120 microns. 5 microns times 30 would be about 150. It's about 120 microns wide by 150 microns high. This is only an estimate, but it's actually pretty close. Let's try another question. How thick is that thick yellow line along the midbrain, MB, over on the right? And I put some blue arrows there pointing to it. Again, we could move our little blue lines over there, but you probably don't need to. And it looks like it's maybe 1/4 to 1/2 of the diameter of a nucleus. So 5 microns times 1/4 is about 1.25. We could round it down to 1 micron. And 5 times 1/2 is about 2.5 microns. So whatever that thing is, maybe a boundary layer of secreted protein and connective tissue around the brain. Anyway, whatever it is, it's about 1 to 3 microns thick. Let's try another one. Example 2 shows the skin of a frog with the outer layer up at the top labeled SG, and in the middle is a mucus gland, MG. This is what secretes the slime. So what's the size of the mucus gland between the two blue lines? And a second question, what's the size of the whole image, the length of the blue arrow at the bottom? Here I'll suggest that you pause and see if you can work it out for yourself. There are some clues in here that are on the bacterial ruler, at least one, and you should be able to work out for yourself the approximate size. For step 1, we can use a nucleus again. There are a lot of dark stained circles or ovals here that are almost certainly nuclei, 5 microns. Notice that some are a little longer and some are a little narrower. My estimate for 5 microns is sort of in the middle. Again, we could line a whole bunch of them up across the middle and say that the mucus gland is probably more than 10 and less than 20. And if that's true, then it's between 10 times 5 microns is 50 microns, not as big as 20 times 5 microns equals 100 microns. It's probably between 50 to 100 microns. What about the width of the whole image? Well, here we can take advantage of the fact that we've already estimated the size of the mucus gland, and the mucus gland is about half the image. That would make the whole image about 2 times 75 microns, somewhere around 150 microns or micrometers wide. Now go on to watch the next video or practice more problems on your own.